Hi everyone, this video will going to discuss about stress marker in human and kinetics of enzyme. The objective of the experiment are to observe the color change of concentration of substrate by the presence of enzyme, to investigate the temperature effect on enzyme activity and to identify the correlation of salivary amylase activity towards stress activity. Stress marker: the higher level of stress, the higher activity of salivary amylase. Substrate concentration: as the concentration of the starch increase, the rate of enzyme activity also increase. Temperature effect: if the temperature increase, the rate of enzyme activities involving all different enzyme also increase. In standard reference, the starch solution were prepared from stock solution of 1.0 mg/ml into dilution of 0.01, 0.025, 0.05, 0.1, 0.3, 0.5, 0.7 and 1.0 mg/ml. All dilutions were added of 1 ml of iodine and each of it prepared for 50 ml. Absorbance of each dilution measured at 590 nanometers. .8 milliliters of 0.01 mg/ml, 0.025 mg/ml, 0.05 mg/ml, 0.1 mg/ml, 0.3 mg/ml, half a mg/ml, 0.7 mg/ml, 1.0 mg/ml were added into different test tube. 8 milliliters of distilled water and 1 milliliter of amylase was added in test tube. All of the test tubes were shaked using shaker until the solution dissolve. Incubate the solution for at 10 degrees Celsius, 3 degrees Celsius, and 4 degrees Celsius, for 10 minutes. In this temperature effect experiment, we can combine with concentration effect experiment which it required of incubation at 35 degrees Celsius as same as temperature effect experiment. After incubation, all test tubes were placed in ice bath. 1 milliliter of iodine was added into the test tube then placed in an ice. After 2 to 3 minutes in ice incubator, all of solution in test tubes were took out. 9 of cuvette were prepared for the solutions. Each of the solution filled into the cuvette. Only 3/4 of cuvette was filled by the solution to be measured using spectrophotometer. Absorbance was measured at 590 nanometers. All of the solution were measured continuously and the absorbance were recorded. For the stress activity, saliva from mouth were collected after rinse the mouth. 5 milliliters of 0.1 mg/ml starch and 1 milliliter of salivary amylase were added in test tube. The mixture was incubated at 35 degrees Celsius for 10 minutes. After 10 minutes, the mixture was took out from incubator and placed into ice bath. After placed into ice bath, 1 milliliter of iodine was added into mixture and shaked it. Absorbance was measured. Stress activity started for 20 minutes. The stress activities include running, push-up and star jump. After 20 minutes, salivary amylase was collected from mouth and steps before stress activity started was followed for after stress activities result. This is the absorbance reading of standard reference. This is graph velocity over substrate. This is the result for concentration effect in enzyme experiment. This is the result for temperature effect in enzyme experiment. Amylase is the primary enzyme in saliva that acts on starch in food and breaking it down into maltose, a smaller form of carbohydrate. So this is the reason why we could detect the taste of sweetness as the maltose is released. In order to understand the reaction of amylase upon starch, we would have to prepare the standard reference, a standard curve of different starch concentration versus absorbance. The plotted graph of standard reference acts a benchmark for us to evaluate the kinetics of industrial amylase towards substrate concentration and temperature effects. Therefore, a method called spectrophotometry, also known as colorimetry, is used to measure the concentrations of specified samples. We didn't get the actual result because of the improper technique of handling the spectrophotometry, but for the sake of learning purposes we have changed the absorbance value of our samples. So, we assume this is our data for standard reference. For salivary amylase activity prior to stress marker in human, one of our group members decided to carry out some activities such as running, star jump and push-ups that could trigger their anxiety disorder, specifically known as obsessive compulsive disorder, OCD. Thus, the absorbance values before and after performing those activities are 0.205 nanometers and 0.309 nanometers respectively. From the achieved values, we could say there is a sudden increase of absorbance value right after the stress-related activity have been carried out. The process that actually happened in our body when the stress level gets high is the immoderate production of salivary amylase takes place. This indicates an increased amount of amylase in order to break down the starch into maltose. Specifically, the starch concentration in our body stays the same for a particular time, but the concentration gets lower when the stress level increases due to high amount of amylase production, resulting more maltose produced. As we can see from the result, the enzyme concentration stays the same throughout the experiment, whereas the concentrations of the substrate, which is the starch, are in increasing order. Once the iodine was put into each of the test tubes, there are a few test tubes that turn into dark blue, light blue, and there are also the ones that do not change at all. The principle behind this occurrence tells us when the concentration of the starch increases, the color of the mixture gets even darker. The actual reason of how the iodine turns the color of the starch into blue is because of the long polymer chains of glucose called amylose contained in the starch. Iodine is known to be insoluble towards water, as the iodine molecules slip within the amylose coil in the presence of potassium iodide. A linear triiodide ion complex forms and causes the dark blue formation. By looking at the Michaelis-Menten graph, as the concentration of substrate increases, the velocity increases too. Meanwhile, the graph of Lineweaver-Burke plot helped us to get the value of max and km, which are 0.012 and 0.833 respectively. The temperature effect using industrial amylase is about how the amylase activity affected by the temperature of 4 degrees Celsius, 35 degrees Celsius and 40 degrees Celsius. Based on the plotted graph of Lineweaver-Burke, amylase has a temperature that works optimally around our body temperature, but in this case, 35 degrees Celsius would consider as optimum compared to the remaining temperature, since the reaction rate 35 degrees Celsius is high, but when it comes to 4 degrees Celsius and 40 degrees Celsius, both of reaction rates are really low and the plotted values located near to each other. This is because when the temperature is too low, the enzyme reaction becomes deactivated, whereas high temperature led to enzyme denaturing and it becomes completely destroyed. In short, we could say that the enzyme activity is strongly affected by substrate concentration and also temperature. Thank you for watching this video. Hope you enjoy it.